Hi, my name is Trevor McQueen. I'm here today to tell you a little bit about our company, Neptune Fluid Flow Systems, and our journey through the Innovation Crossroads program. Pharma has a problem. Half of their R&D dollars, that's right, fully half of their R&D dollars are wasted on irreproducible science. And this boils down to a lack of reproducibility, repeatability, and reliability in the underlying scientific techniques. In one of those techniques, protein structure determination, we have developed an innovative solution. And that solution is to take advantage of advances in microfluidic technology to prepare samples instead of in multiple discrete steps, do it all in a single concerted step on a chip. And by doing this, we go orders of magnitude faster, and we eliminate all of the transfer steps, which are notorious for destroying proteins during them. This allows us to have a vastly higher reproducibility, repeatability, and reliability in our core technology than anything else out there on the market. And if you compare it to what's out there on the market, we stand head and shoulders above it. We are way beyond it. And the market is big. In 2019, pre-pandemic, it was $8.5 billion and had been for the last 5 years, averaging a 70% year-over-year growth. It's gigantic and growing. But you'll notice something interesting about it. The majority of this industry is academia. And that's because pharmaceutical companies have outsourced this portion of the drug discovery process to academia due to the lack of reproducibility, repeatability, and reliability that they discovered when they tracked what was happening with their scientific budgets. And so when we started Innovation Crossroads program, we're like, wow, big market, growing fast, we're going to be an instrument sales company, just like everything else you'd see. We're going to produce an instrument, we're going to sell it for $750K, that's it. And we looked at the market. The market is quite crowded. There's over a dozen different technologies out there. The primary biggest ones that are out there most successful right now are shown here on the slide, but there's many more that we couldn't even fit on the slide. It's a very crowded marketplace. And so, thanks to Innovation Crossroads, we thought outside the box. What if we became a CRO? Well, the CRO landscape is very non-competitive. There's exactly one CRO for cryo-TEM in the entire country. And they have two locations, one on the West Coast, one on the East Coast. Just so happen to be directly across the street from Merck Labs because 2/3 of their projects are done with Merck. And however, the market as itself is much more spread out. And there's a whole Midwest segment that is entirely unserved in the CRO model right now, but there's a lot of pent-up demand for it, as there's almost as many in the Midwest as there is on the entire East Coast. And so our plan is to take our new technology, which delivers more reliability, more repeatability, and more reproducibility, and deliver that to customers in a CRO model, thereby saving them a lot of time and in the end costing them an order of magnitude less to get that protein structure. So our go-to-market strategy is really target this service model segment because it's the easiest to enter, it has the least competitors, and it is a large market. It's a $200 to $400 million potential market looking at the number of customers that nanoimaging currently serves. And then we'll keep our eyes out on the direct instrument sales, but if that threatens to eat our service model's lunch, we may or may not pursue it. So our timeline is in the next 2 years, we're going to keep developing this CRO model that we already have, and we're actually going to start monetizing it as best we can to see how large we can grow this, how rapidly. And then in a few years, so 3 to 4, we'll circle back and we'll reappraise whether or not to pursue the direct instrument sales model or to pursue a strategic partnership where we become effectively a service provider through one of the big microscopes. So they sell the microscope and we then are the contractor who serves to help them prepare their samples to run on the microscopes. Now our team is primarily myself and my co-founder, Dr. Winnie Liang. And when we first started the Innovation Crossroads program, it was just us. But through the Innovation Crossroads program, we've got a wonderful board of technical advisors and a ton of business mentors, all of whom have been absolutely critical to our growth and development as a company. As you can see, they are the ones who pushed us to think, what if instead of being a direct instrument sales company, what if you became a CRO? And through that, they have radically changed our business model and helped guide us towards a much brighter future. And that future is what we're looking for is in the next 6 months, we're going to start raising a $1.5 million round. This is so that we can lease and then own our own cryo-TEM that we can then sell time on in order to become a true proper CRO. And we've done the math, we've got the quotes and everything, and it takes about 30 samples a year in order to break even on the cryo-TEM service side. And we are already preparing more than 30 samples a year, which means we should be able to directly scale and get a lot of revenue quickly. And finally, if any of you have any connections to any company or group that is interested in cryo-TEM, so drug discovery, protein structures, lipid particle structures, any biological structure determination, please do not hesitate to send them our way. We will absolutely help them prepare their samples better and get better data quicker, cheaper. Because the more they know, the quicker they can deliver new drugs, new therapies, and save lives. Thank you. Any questions?
Yeah, Trevor, this is Jonathan. Let's just back up a minute. If you'll indulge me, please, to repeat, what technology do you have and why is it great?
Yeah, so what we have is, so to tell you a little bit, let's start first with cryo-TEM. So in order to do cryo-TEM,
Which means?
So it's cryogenic transmission electron microscopy. This is how they currently take pictures of proteins. So literally take atomic resolution pictures of a protein molecule and then figure out where every atom is placed and therefore determine the structure of the protein. This is the first step in the drug discovery pathway, because what they're looking for is they're looking for pockets and regions that they can attack with a drug molecule and thereby develop a drug that targets that specific protein to either knock it out of action or to enhance its action, depending on which way they're trying to go. Now, the first step is you have to get your protein to cryogenic conditions, and you can't freeze it into ice. If it freezes into ice, then the ice crystal will shred your protein, and you'll be left with just random atoms and amino acids just floating around, and you won't have your protein. You'll have destroyed it. So you have to do a process called vitrification. So this is ultra rapid cooling, such that instead of freezing, the water vitrifies and it turns into glass, just like in my glasses and in your glasses, right? That's an amorphous solid. And so if you cool the water, which the protein is in, fast enough, you can get to that vitreous solution. Now, this is currently done in a 5-step process. We, however, via a microfluidic deposition, do it in 1 step. And this allows us to take, instead of taking around 2 minutes to do this process, it takes us 86 microseconds. This allows us to outrun a lot of kinetic processes. In fact, our proteins don't have enough time to aggregate. They cannot come together because they don't have the time to move. Further dissociation, they don't have the time to break apart. A lot of the unfolding steps take on the order of milliseconds. And again, since we're on the tens of microseconds scale, we outrun most of these biological damage processes, giving us much more reliable and reproducible results with much better protein structures.
Thank you.
Well, I love that because that is what I do. So yeah. Tell me something. I mean, I love this. I actually well, way long ago worked actually with Yale company on the first equipment actually for cryo-electron microscopy. Not there yet, but we've been working on viruses and so on. And then I continue my career doing drug design. And yeah, I do that. So so tell me something. Have you been able to solve one structure using the technique?
Yes.
Okay.
Well, we actually did this. It wasn't with a classic protein, so it was with a nanoparticle embedded lipid. So effectively, it's one of the drug delivery devices that is prepared by a company here called T&T Scientific, and it's actually used in I forget. I think it's Pfizer's COVID vaccine involves.
Yeah, the lipid nanoparticles are both. I mean, Pfizer and Moderna are using lipid nanoparticles.
And
but
so,
the ones that you're referring probably is on BioNTech. Pfizer.
Yep. Yes. Okay. Yes. BioNTech.
Okay. So, but I'm referring to proteins because this is the big thing of the deal, and the rheological characteristic of the protein is completely different than the liquid nanoparticles in the nanotubes. So no, I mean, I'm telling you because that could be a major source of a show here, and I think that to demonstrate actually the this MVP, like you can call it, you should go there, talk to somebody on cryo-EM, bring your stuff, and do it. And do it for a protein. I mean, do it for a big protein, like a membrane protein or something like that. That is the target for cryo-EM.
No, this is, please do not spread this around, as we've just prepared the samples and we're not entirely sure whether it'll work out in the end.
No, that's okay. That's okay. But that's okay. I would not spread it around. But the point is that this is.
We prepared the COVID spike protein for a research group at Stanford. We have actually sent it to them. They should be receiving it tomorrow. And then over the weekend, in theory, a graduate student will be running these prepared grids. And then in theory, if all works well, by the end of next week, we will have a solved structure for the COVID spike protein.
Perfect, because if you do that, actually, the vendors of EM will come to you. So as soon as it's there, they will come to you. So you just relax, sit down, get a margarita, they will come to you. So because this is the problem, the big problem these days. I mean, the reproducibility of the jet flows machine and all that to get a cryo-EM. I'm not sure, because the things that we are solving are big. And big in a nanotube or big in a lipid nanoparticle is okay, because they are very symmetrical. Big in a protein is a hell of a mess because it's full of water. Lipids don't have water.
Oh, absolutely. The reason why we actually started with the lipid nanoparticle was we are working with TNT Scientific to make this a standard quality control metric.
Yeah.
So you literally take a picture of the batch. So you can show that each batch, the lipid nanoparticle really is in the same structure as what you say it should be.
Awesome.
It allows them to charge a higher price on to BioNTech. That's we're trying to get into a sort of assembly line kind of start.
Great. And do we have any more questions from the panel?
Hey, Trevor, how do you keep your focus if you become a CRO?
Well, that's where we hire more people. So effectively what we currently have is I'm running around, I'm doing the R&D, I'm doing the development of the microfluidics, I'm doing all of the testing, I'm actually forming the collaborations with the professors. So I'm going out and talking to professors, I'm finding out their proteins. To be honest, they tell me everything about their protein, it goes over my head. I just need to know general conditions. And then we work with them to prepare it and help them learn. So as of right now, I'm close to my limit. And once we start truly doing the CRO, I will absolutely need people to run this. But the wonderful part about that is once we truly are the CRO, we can start specializing. Because then there are clear roles. There's roles in terms of getting more customers in. There's roles in terms of getting more data run, so more samples prepared and vitrified. There's also the role for running the microscope, right? There will be someone whose job is to run the microscope and take all the samples prepared and just check, right? And that will be their one role, and we will have enough for them to keep doing this. Especially once we lease our own microscope, we'll have dedicated time, which we can use to do a lot of work, because there's a lot of unknown proteins out there that if we can show we can do just a couple of them, as Rolando was saying, that puts us, people will come to us at that point because we can do something no one else can. So we are preparing for that. We are in fact hiring our first employee. So we're gonna try before we buy. So start them out as an intern, and then if they succeed in the next 10 weeks, we will offer them a full-time position. And that will be to run the microscope here at Giant. So collect the data for us.
Thanks. That's all I have.
Congratulations and thank you.
Thank you.